Hello, everyone. Thanks for coming. Wonderful to see such a turnout. Not surprising, because we've got Danica talking, and Danica, as is well known to many of you, Danica's been at WeHi for 3 years in my lab. I'm very fortunate to have had Danica as a student. She's achieved a lot in her time, and she's been very active in a number of areas in and around the Institute, has gone out and done extracurricular stuff. Just a testament to her energy and enthusiasm. I think Danica's returned a really very interesting and distinctive PhD in bringing together things that we've been working on for many years and advancing them in quite a distinctive way. I'm not going to say any more about the content because she's here to deliver it herself, but just to say that, well, we'll leave it there and we'll take comments at the end. Danica, tell us all about identifying novel functional correlates of acquired immunity to malaria.
Thanks, Louis. So my project's really focused on trying to understand acquired immunity to malaria and using human population studies and developing some new tools to measure these immune responses in vitro, to really try and identify what immune responses are important to malaria and which ones correlate with protection from disease. So although a lot of the global mortality and morbidities in the African region, I wanted to focus on something a little bit closer to home, and that is malaria in our region, in South Asia, Southeast Asia and the Western Pacific, which, according to recent estimates, puts 2.3 billion people at risk of malaria each year. And this is 2.3 million cases. And in Papua New Guinea, which is the focus of my research today, it's 150,000 cases per year. And although only a proportion of these lead to death, you can still see that it's a really big public health burden in these countries. Of the 6 different species that can infect humans, the focus of my talk today will be Plasmodium falciparum, which contributes to the majority of the deaths associated with malaria. So the way the immune system responds to the parasite differs throughout the parasite life cycle. So here's the life cycle for Plasmodium falciparum, and it starts with the bite of an infected mosquito, which injects sporozoites into the skin, which quickly find their way into the bloodstream. And it's during this stage that antibodies can block their invasion into the liver. And then once resident within the liver, these parasites can develop, and it's at this stage they're targeted by CD8 T cells and also some innate-like cells. When the parasite leaves the liver, these merozoites can go on to invade red blood cells. And then mature to form a schizont, which then ruptures to release merozoites. And this is the asexual blood stage cycle. And it's during the blood stage that antibodies, CD4 T cells, and then pro-inflammatory cytokines can play an important role. Antibodies can also play a role in blocking transmission, so by inhibiting fertilisation within the mosquito midgut. So the majority of all of the malaria symptoms are associated with this blood stage of the parasite life cycle. And these different immune mechanisms have mostly been identified from rodent malaria models, but the precise roles that each of these different mechanisms play in human disease is really less clear. So people living in malaria-endemic countries can develop immunity to malaria, but it requires multiple malaria episodes. And it results in something that's species-specific, in that immunity to Plasmodium falciparum does not protect against Plasmodium vivax. And unlike most other infections, doesn't result in sterile protection. But instead, you have parasites, but you're able to control the burden of these, and you have reduced clinical symptoms. And it's still unclear about whether or not ongoing exposure is required to maintain this immunity. So during the talk today, I'll be using 2 terms quite frequently. The first is clinical malaria, and this is when you have infection that results in fever and symptoms. And I'll also be using the term clinical immunity, which is the presence of infection without these fever and symptoms. So this ability for people naturally exposed to malaria to develop immunity really underpins the rationale for developing a vaccine against malaria. However, we really need to understand what the protective antigenic targets of this immune response and which of the immune mechanisms are important to really guide better vaccine design. So when someone's developing clinical immunity to malaria, there's a range of different factors that can influence this development. I've sort of broadly classified these into factors of the human host, malaria parasite, and some more environmental factors that can influence disease prevalence. And these things are challenging for both the person to develop immunity, but also are important for when researchers are trying to study acquired immunity. So in the human host, there are certain genetic factors, such as red blood cell polymorphisms, that can influence the ability for people to be infected with malaria, but also some other things like cytokine gene polymorphisms that predispose people to clinical disease. Comorbidities are really important in malaria, in particular anemia, HIV co-infection, and helminths can influence the way the immune system responds to the parasite. And the age of an individual and how mature their immune system is can also influence the development of immunity. On the parasite side of things, Plasmodium falciparum has extensive genetic diversity and also variation in the antigens that it presents to the immune system. There's also evidence that the parasite itself can cause immune modulation, either by inducing regulatory type mechanisms or by impeding the development of memory. Onto the more environmental things, the amount of prior exposure has a really important influence on how much immunity has been acquired. And as I'll show you today, in these endemic settings, the age of a child and the amount of prior exposure are really tightly linked. When assessing immunity in the lab, we need to consider whether or not someone has a recent infection or a current infection. And I'll show you a little bit more about that in my talk today. Also, whether or not there are multiple species. So, for example, a co-infection with Plasmodium falciparum and vivax can make it difficult to determine which of these species is causing the clinical disease. And there's also often many mixed infections where multiple different strains of Plasmodium falciparum can contribute to the disease. And then two factors that really affect your risk of being infected. So malaria control measures such as sleeping under a bed net or access to drugs can influence how often you get clinical disease. And also the transmission intensity, which really comes down to the risk of being reinfected. So when we do these human population studies, we really need to take all of these things into account to get an overall picture of the development of clinical immunity. So by testing different immune responses in vitro, what we're able to assess is whether or not something correlates or associates with immunity. And you'll hear me using these terms a lot today. As sort of by this strategy, it doesn't give sort of definitive proof, but we hope by testing these various different immune responses in multiple different studies, we get much closer to identifying what is truly important in the immune response. So, most of the traditional approaches to understanding immunity to malaria have focused around ELISA-based serology, where different malarial antigens are expressed generally in E. coli systems and coded onto plates, and then antibodies are added and the assays sort of measured to saturation endpoints. However, there's a greater understanding that we need robust functional assays to better aid vaccine development. And these functional assays really need to represent credible immune mechanisms. So when we're talking about acquired immunity to clinical disease, this really focuses in on the blood stage. And we believe the different credible mechanisms are antibodies that can inhibit invasion or opsonize parasites, and also CD4 T cell effector functions. And I'll be talking about opsonization and CD4 T cells in the talk today. But on top of just developing these robust assays, these assays really need to be tested for whether or not the immune response we measure is associated with protection. So as I mentioned, Plasmodium falciparum has extensive genetic diversity. And this is both in the different allelic diversity and genetic polymorphisms. And this creates not only a challenge for the individual to develop immunity, but also creates a challenge for studying this immunity in the lab. So it's going to be really, really important that addressing antigenic diversity is taken into account when designing vaccines. However, a lot of vaccines are designed based on one reference sequence. And quite commonly, this is the 3D7 lab parasite strain, which has been growing since the 1970s, but was the first parasite to be sequenced. And so it's really not quite clear whether or not these sort of selected lab strains are really a suitable representation of the genetic diversity in locally occurring parasites. And this is because various different immune mechanisms have shown to be strain-specific. This is especially pertinent for the protein PFMP1 on the surface of infected erythrocytes, which has has shown strain specificity in both the T cell and the B cell immune responses that are elicited. Growth inhibitory antibodies have also shown strain specificity, and this has also been seen at the level of individual antigens, where things like merozoite surface protein 2 and 6 have different allelic forms and display strain specificity. So a lot of the focus of my research has been on the merozoite form, the parasite, and just as a reminder, that's the form that bursts out of a schizont and then can reinfect red blood cells. And merozoite antigens are attractive vaccine candidates because there's that extracellular step, so they're exposed to the immune system. And most of the antigens on the surface of the merozoite are immunogenic. So here's just a schematic of all the different proteins that are exposed to the immune system, both present on the merozoite surface and also released from these intracellular organelles, the ruptures and the micronemes. So there's really a lot of different proteins that are targeted by the immune system. But as I'll show you today, just because antibodies can be elicited against these proteins doesn't necessarily mean that those antibodies are protective. And just to illustrate this, although there's been decades of work looking at various different proteins, two recent groups have taken large panels of different proteins, both E. coli and mammalian expressed, and looked for associations with protection to find novel vaccine candidates. And you can see here this is an African study, and this one here is one of the PNG studies I'll be talking about today. And I just want to illustrate that although they're looking at responses to similar proteins, there's a lot of discrepancy in which studies report associations, which is a really challenging aspect of malaria immunity and why that particular findings need to be replicated in multiple different transmission settings. And it's also very unclear what these different antibodies do. But despite this, there is a lot of interest internationally to target this blood stage for a vaccine. So here I've just pulled up a list of the current blood stage vaccines in clinical trials. You can see many of these are merozoite surface proteins. And it's really unclear what a lot of these different antibodies do. But I think this nicely demonstrates just how much effort is going into generating a vaccine against the blood stage. So in my talk today, I'll be talking about opsonizing antibodies to Plasmodium falciparum merozoites, focusing on how I developed and validated a phagocytosis assay, how we correlated these responses to clinical outcomes, and some of our investigations into strain-transcending antibodies. I'll also be talking about some CD4 memory T-cell responses to Plasmodium blood stage parasites, how I developed a re-stimulation assay, and some of our work looking at comparing malaria parasite responses to cytomegalovirus. So firstly, looking at opsonizing antibodies. So we have a really clear evidence that antibodies can protect against disease. And this was shown over 50 years ago when immunoglobulin was isolated from hyperimmune African adults and transferred into children suffering from severe disease, and it was able to alleviate their symptoms. As I've shown you, merozoite antigens are strongly targeted, but it's really unclear what these antibodies are actually doing to protect the host from malaria. So when I think about what these different merozoite antibodies can do, they can really do two diverse functions. Firstly being functions that are direct, so the antibody is able to bind to the merozoite and block invasion, etc. Or opsonization, which is where antibodies bind to the surface of the merozoite and then can trigger effector functions by the Fc portion of the antibody. So firstly, direct functions, these can be invasion inhibition, agglutination, or perhaps complement fixation. But the focus of my talk today is really on these opsonizing antibodies, which in vitro have been found to produce respiratory burst from neutrophils, which is the release of reactive oxygen species that kills bystander parasites. Antibody-dependent cellular inhibition, which is from monocytes and the release of an unknown soluble factor that, again, kills bystander parasites. Or phagocytosis, which is the engulfment of merozoites. And all three of these mechanisms are mediated through Fc receptors. So this respiratory burst from neutrophils has been shown to associate with protection from clinical malaria. However, high production of reactive oxygen species has also been linked with severe malarial anemia. So whether the protective and pathogenic roles for ROS is really unclear. This ADCI assay has been around for decades and has been used to identify new vaccine candidates. But it's been untested about whether or not this is relevant for protecting. And as I mentioned, it's an unknown soluble factor, so it's really unclear whether or not this is even relevant in vivo. Phagocytosis has been observed for many years, but hasn't been not tested for associations with protection. So as I mentioned, all three of these require Fc receptor interactions, and it's likely that opsonizing antibodies can trigger potentially all three of these mechanisms. And it's likely that phagocytosis is the first step in all three of these as well. So my research questions have been, are merozoite opsonizing antibodies associated with clinical immunity? Are opsonizing antibodies strain-specific? And to try and understand which antigens might be targeted by these opsonizing antibodies. And to do this, I used some plasma samples from a treatment reinfection study from the north coast of Papua New Guinea back in 2004, where school-aged children were enrolled, a plasma sample was taken, and then children were treated with anti-malarial drugs to clear infections. And then they were monitored for reinfection and clinical disease over the period of 6 months. And this is just to show how useful this study has been for understanding naturally acquired immunity and the epidemiology in Papua New Guinea. And so it's a really highly powered study to answer some more technical questions. And this is because during the 6 months of follow-up, 93% of children were reinfected by Plasmodium falciparum, and you can see this here. But 50% experienced a clinical episode. So you can see that there. So this really allows us to sort of ask questions, what is it about the children that were reinfected but didn't present with clinical malaria? Because these are obviously showing clinical immunity. There was 68% of these children were PCR positive for Plasmodium falciparum when the plasma was collected. And I'll refer to this as baseline. And this is really important for its effects on the antibody response. So to assess these opsonizing antibodies, first needed to develop a new technique. And this was because existing techniques used a really poor quality of merozoite and relied upon primary monocytes, which of course have purity issues, are poorly reproducible between assays. But I think the most important thing is these monocytes have lots of complexity in their own biology and can phagocytose merozoites in the absence of antibody. So to get around this, designed a new system where we could make high-quality merozoites and move to a more cell line-based system as a robust way to just measure the effects of the antibody. And this allowed us to also increase the throughput. So to do this, first magnetically separate the trophozoite parasites. These are then treated with a protease inhibitor for 8 to 10 hours, which prevents Charcot rupture and yields these nice membrane-enclosed merozoite structures. These can be filtered to release merozoites and hemazoin crystals. And because hemazoin is known to be highly pro-inflammatory and influence phagocytosis, this is removed by magnetic separation. To help visualise phagocytosis, the merozoites are stained with ethydium bromide, which intercalates into their DNA. And then these merozoites are counted by flow cytometry, as they're only a micron. Then they're opsonized with diluted plasma. Here I'm showing the Australian negative control and PNG plasma. And then phagocytosis is by addition of this THP1 monocytic cell line for 40 minutes. And the utility of this cell line is firstly, it's non-adherent, so it's really great for flow cytometry. But second is this cell line will only perform phagocytosis in the presence of antibody. You can see that here. In the presence of malaria-specific antibodies, there are merozoites in the cytoplasm, and this reads out as a proportion of the THP1 cells becoming fluorescent as they've taken up a labelled merozoite. So this has allowed us to develop a technique that we can screen several hundred plasma samples in duplicate in a single experiment. So it's been really useful. However, because this is my completion seminar, I will say that this particular technique is not amenable to a social life. So we validated this assay via a range of different methods, and I won't go into that today, but I just thought I'd show some nice time-lapse images that nicely depict that this is indeed phagocytosis that we're seeing. So firstly, here with the non-exposed plasma, we have the monocytes, the THP1 cells in grey, and the little red blobs are the ethydium bromide labelled merozoites. And as I mentioned, they're non-adherent, so everything moves around in solution, but you can see that the cell behaviour doesn't change very much. However, here with the PNG exposed plasma, you can see the cells start sending out pseudopodia and performing phagocytosis. and the ethidium bromide signal remains within the cytoplasm. So we're very confident that by measuring the ethidium bromide signal, we are actually indeed picking up phagocytosis. As I was validating this assay, I was surprised to find that merozoite phagocytosis requires only very low antibody concentration. And this was surprising because the other existing functional assays in malaria, growth inhibitory assays, and phagocytosis of infected red blood cells, typically use a 1 in 10 dilution. However, in my hands, the assay was saturated out to a 1 in 5,000, and it ended up selecting a 1 in 120,000, which is here, to resolve high versus low responses, which I think tells us two things. Firstly, that the process of merozoite phagocytosis is very efficient, but also just goes to show just how much antibody these young children in PNG have circulating. As I mentioned, we were interested in seeing whether or not these opsonizing antibodies are strain-specific, because many of those other functional mechanisms have been shown to display strain specificity. We were interested to see whether or not these lab strains are a true representation of locally occurring parasites. And this is really to understand whether or not merozoite opsonization is a robust immune correlate, as many of these assays show high degrees of variability between parasite strains tested. And to do this, used a variety of lab strains. You can see here, these are the sort of original geographic origins of many of these parasites. However, they've been in culture for some of them since the 70s. And I think just goes to show maybe how far removed they are from their original areas of origin. For example, many of these different parasites were cryopreserved before I was born. However, with the PNG isolates that I was able to adapt with the help of Danny Wilson, these are from the local, from the children in the study. So they're obviously quite representative of locally circulating parasites. So to do this, we used red blood cells that were cryopreserved from high parasitemia infections at the baseline bleed. And I guess the reason why people use lab strains is that parasites from peripheral blood generally don't adapt well to the lab, and they're difficult to work with. But nonetheless, attempted to adapt these parasites and made 2 attempts for each of the 30 and managed to get 12 lines that were able to stably grow. By performing some genotyping, found out that 7 of these were single clone infections, whereas the additional 5 were 2 different parasite strains. So you can see that here. These are the 12 different strains that I got growing. 3 of them were lab adapted by Danny Wilson. And you can see they sort of vary in how long it takes them to adapt. And as I mentioned, some of these were multiple infections. And then by limiting dilution, was able to clone 3 lines successfully. So that gave 10 to work with. We were quite surprised to find that by doing this MSP2 genotyping, that all of the parasites were unique in that they didn't share any similarity in this MSP2 genotype, which I think tells 2 things. Firstly, just how much local diversity there is in the parasite population. But as this merozoite surface protein 2 is also a likely target of the opsonizing antibodies, again, just sort of demonstrates how much diversity there is in these merozoite surface proteins. So when I tested these opsonizing antibodies against some of the outcomes, you can see here this is for 3D7 merozoites. We found that older children have much higher levels of antibodies, which is consistent with what we know about immunity in that it's acquired with age. There was also a location effect in that those children that lived inland had higher levels of antibodies. But we know from the epidemiology that these children inland have higher levels of exposure. So as I'll mention later in the talk, when I perform the statistics, we adjust for age and location. But a range of other demographic effects like sex had no effect. As I mentioned at the beginning, 70% of these children had an infection at their baseline bleed. And as you can see here to the response to 4 different lab strains, that in children that had an infection, they had much higher levels of antibodies, which is consistent with what we know about active infection boosting antibodies. But as I'll talk about in the subsequent slides, it's known that these antibodies that are boosted by active infection have a really short half-life that can range from a fortnight out to a month. So we were surprised to see the extent of this boosting, but we think that these antibodies are likely very short-lived. So then when we related these opsonizing antibodies to clinical outcomes, as I mentioned, there were some that were protected from clinical episodes, and there was up to 3 episodes of clinical malaria in 6 months. And you can see that in those that are protected from clinical malaria, they have high levels of opsonizing antibodies relative to those that succumb to multiple episodes. But we only saw this effect in those that were uninfected at baseline, as those that had an infection had much higher levels of antibodies, and they really showed no relationship to the clinical outcomes, which again suggested to us that these boosted antibodies likely don't remain in circulation for very long. So if you measure a high response at baseline, this doesn't really read out to protecting you during follow-up. So this is just to show the responses for the 15 different lines that I tested, and that we saw this statistical trend in the uninfected individuals, but never in the infected. So because I did this against the 15 different lines, we could see how the responses correlated to one another. And it's a very complex matrix, but I just draw your attention to this top line here, which I think nicely illustrates just how tightly correlated these responses are, and indicated that there really was no difference between lab strains and P&G lines, both in how they related to one another and together, which sort of gave us the first hint that there seems to be very little evidence for strain specificity in these merozoite opsonizing antibodies. So to assess whether these responses are associated with protection, we can use 2 different statistical methods. The first, looking at whether or not there's an association with reduced incidence of clinical disease, and this is using Poisson regression modelling and an incidence rate ratio. And here's just a schematic for how that reads out. So if it's above 1, that relates to an increased incidence. At 1, there's no change. At 0.5, a 50% reduction. And down here at 0, that's a 100% reduction in the incidence. For each of these, I'll be showing a 95% confidence interval. And in general, if the 95% confidence interval is lower than this dotted line that I'll include at 1, then this is a significant association. The other way we can assess things is looking at whether or not there's a delayed time until clinical disease. And this is using Cox regression modelling, or a hazard ratio. So here is just an example of a Kaplan-Meier plot that I'll be showing. And as I mentioned, there's that spatial confounder where, depending on how far away you are from the coast, influences the transmission intensity. And you can see that here, for the children that live further inland, they have a much higher risk of developing disease. So when I report these associations, I'll show them unadjusted, but then when I use multivariate models to adjust for the distance from coast, but also the age of the individuals. So when we did the Poisson regression modelling against all the different lines, you can see that these opsonizing antibodies broadly associate with reduced incidence of clinical disease. And this sort of ranged from 52 to 93%. And although there was some variation across the different parasite lines in terms of the absolute estimate, they were broadly protective. And many of these remained significant after adjustment. We were quite surprised to see that 2 very typically used lab strains, 3D7 and D10, were not protective in this cohort. So because we'd measured these responses against all these different strains, we could look at combinations of responses. So firstly, looking at how people responded to all the lab strains, all the P&G strains, and then all the strains together. And what this played out when we did these combined responses was that these opsonizing antibodies were really broadly protective, resulting in an 80% reduction in incidence. But again, this was only for those that were uninfected at baseline, as when we looked in those that were infected, we saw no association with immunity, which again is indicative that these boosted antibodies are likely really short-lived, and hence if you measure them at baseline, they're not an appropriate measure of immunity. As I mentioned, there was the 2 different ways to assess immunity and risk. So this is the second way, looking at the time until clinical disease. And this is here just looking at the aggregated responses across all the different lines. You can see that high levels of opsonizing antibodies result in associate with an 81% reduction in risk. But again, this is only in the uninfected individuals. In the infected individuals, we see no association. So I've shown you that opsonizing antibodies increase with age, they're boosted by active infection, and they're really influenced by the intensity of transmission. We saw no associations to clinical immunity, in those that had an active infection. But when we measured it in uninfected individuals, where we can measure a steady state antibody level, these were highly protective, resulting in an 80% reduction in incidence and an 81% reduction in risk of clinical disease. And I think it's fair to say that these numbers here are some of the most striking correlations with protection that has been seen in this study and many other studies of protective immunity. So we're quite excited about opsonizing antibodies as an immune correlate. And we're also happy to see that our findings have been replicated in a recent study from colleagues at the Burnett, where they tested opsonizing antibodies in Kenyan children and found a very similar 80% reduction in the risk of clinical disease. But we saw that the responses were highly correlated across parasite lines, which got us sort of thinking about how these antibodies might be strain transcending. And this led us to come up with two different models. The first being that opsonizing antibodies are acquired in a strain-specific manner, in that with each infection you acquire antibodies specific to the various different versions of the merozoite surface proteins that are presented, and then with each cumulative exposure you develop these strain-specific antibodies. But by the time these children are 5 to 12, what you measure in the blood is a mix of lots of different antibodies that can sort of broadly react against lots of different parasite strains. The second is that there are conserved antigenic targets, and that with each infection you get increased numbers of these conserved antigenic targets, which again, by the time you're 5 to 12, you've got a large pool of these antibodies, and then it really becomes whether or not someone has a high response or a low response that reads out with protection. So from some of the mosquito work that was done in this study, it was estimated that these children have 35 infective bites per year. So at the age of 5 to 12, they've had between 175 and over 400 Plasmodium infections, which means that teasing apart these two things becomes really complicated in this age group. But it's quite important that we understand whether or not it's Model 1 or Model 2 if opsonizing antibodies are going to be used to guide vaccine design. As if it's this, then a vaccine strategy will be really useful, but if it's this, then sort of addressing that strain specificity is going to be really challenging for vaccine design. But so testing Model 1 might require looking at antibody responses in first infections, particularly in young children. But we can do some hypothesizing about what some of the conserved antigenic targets might be to look at Model 2. So this got us thinking about the MSP3 family, which, so here's just a reminder of all the different proteins on the surface which may be targeted by this immune response. But we've sort of focused here on the MSP3 family, which shares conservation in its C-terminal domain. And today I'll be talking about MSP3, 6, and MSP-DBL1. So when antibodies were affinity purified by Chris Chu to some MSP-DBL1 full-length protein generated by Clara Lin, when I tested these antibodies, I was able to show a dose-dependent increase in the amount of phagocytosis, indicating that MSP-DBL1 antibodies can trigger phagocytosis. In a recent paper by the group at the Burnett, they showed that purified MSP3 antibodies can induce phagocytosis. But just because you can elicit a response doesn't necessarily mean that these particular antigens are the dominant target of this response, and it definitely doesn't give us any information about whether or not this response is protective. So to address that, I was able to use some knockout parasites generated by Alex Uboldi in Alan Cowman's lab to MSP3, MSP6, and MSP-DBL1. And what I saw when I tested this against a range of different PNG plasma, that I saw no differences in the percentage of phagocytosis between the wild type and knockout parasites, which indicated that loss of any one of these particular antigens doesn't impact opsonization. And the possible explanations for this could be that none of these proteins is the definitive opsonization antigen. But what's more likely is that there are multiple antigens that are targeted by this opsonizing response. But it is possible that this MSP3 family, which shares that conserved C-terminal domain, that it's antibodies to this conserved C-terminal domain, which have been shown by serology to cross-react to other MSP3 family proteins. So we're interested in assessing whether or not this conserved C-terminal domain may be that sort of strain-transcending target of these opsonizing antibodies. So in summary, I've shown you that merozoite opsonization is a robust correlate of protective immunity, and we've replicated this in field isolates and across a range of parasite strains, and it's shown high reproducibility. Opsonizing antibodies are strain transcending but highly protective. And this is because we've seen no evidence for strain-specific opsonization in these 5 to 12-year-olds. And that loss of MSP3, 6, or an MSPDB1 does not affect opsonization. But what's going to be quite important for the field is we've really generated a high-throughput assay that's amenable to large cohorts and vaccine trials, which has really been a limiting factor in a lot of these other assays of functional immunity. Future directions, looking to investigate strain specificity in younger children or some controlled human malaria infections, and looking at this conserved domain of the MSP3 family to look at whether or not we can see cross-reactive antibodies. So in the last 10 minutes, I'm just going to talk about some of the work we've been doing looking at memory CD4 T cells and responses to blood stage Plasmodium falciparum parasites. So memory T cells can be generated when naive cells respond to a pathogen and proliferate and produce effector functions that are able to clear the infection. And so when the pathogen is cleared, many of these cells will die, but what you're left with is a pool of memory cells. And throughout the talk, I'll be using some of these acronyms, which respond to the various different memory populations. And then when these cells re-encounter the next infection, they can proliferate and perform their functions much more quicker and more robust, and are able to prevent the infection or the disease. So most of the work looking at memory T cells has been performed using viral pathogens, and mostly CD8 and a little bit of CD4. But their role in malaria is a lot less clear. And it's important to mention first that red blood cells don't express MHC class 1 or class 2. And so responses to both infected red cells and extracellular merozoites revolve around these antigens being presented by antigen-presenting cells. So it's not a CD8-mediated immunity. And so these CD4 T cells, their pro-inflammatory cytokines that they produce can activate phagocytes to induce parasite killing, and are also very important for providing B-cell co-stimulation. Many have suggested that malaria suffers, that immunity to malaria suffers from a memory failure, and this is based on two really indirect observations. The first being that immunity takes a long time to develop. But I think, as I've shown you today, that degree of genetic diversity and just the amount of different antigens that the immune system has to respond to, I think we believe accounts for this delay in the acquisition of immunity. The second being that malaria infection can reduce responses to vaccination. So, for example, vaccination with tetanus or Salmonella typhi, which is sort of very indirect evidence, but sort of suggests that if the infection reduces responses to these vaccinations, then likely the infection inhibits the generation of memory to malaria itself. But it's really important that we understand more about whether or not these memory T cell responses are a correlate of protective immunity. And to understand, really, is there any basis for this memory failure to malaria? And so this is sort of really hasn't been that well addressed in the field, because most people have relied on peptide studies, where they've chosen peptides to an antigen of interest and re-stimulated PBMCs for many, many days in the lab. And this results in a really low frequency of response, and we've seen that in our hands as well. And part of this is due to the HLA diversity in MHC class 1 that makes these peptide approaches really complex, in that you need really large peptide libraries to cover that HLA diversity. But it's also due to that we really don't know what are the major T cell antigens, so it's hard to know what to start with. And they haven't been assessed for protective correlations. So there's been a change in the field to using whole parasite approaches, but this hasn't been looked that extensively in CD4 memory. But a recent paper came out just this year in a cohort of Ugandan children in a really sort of staggeringly high transmission rate, in that these children have, at least by the mosquito estimates, more than one infection per day. And they found that TNF production was associated with symptomatic infection, but failed to identify a protective correlate. And they didn't investigate the memory response that thoroughly. So we're interested in sort of extending this analysis to a different study in Papua New Guinea, another treatment reinfection study that was from 2009, where 500 children were given anti-malaria drugs to clear infection, and then 2 weeks later, a venous bleed was taken. And so dissimilar to the Mughal study, there are no infections at that baseline bleed. And peripheral blood mononuclear cells were cryopreserved in PNG, and we've used these as a source of T cells in the lab. And so during the 9 months of follow-up, 166 individuals were reinfected with Plasmodium falciparum, and we've focused on these as we can confirm that they had exposure to the parasite. And of this 166, 58 individuals experienced clinical malaria, while 108 remained asymptomatic. So we're really interested in understanding what is the difference between the asymptomatic and those that had clinical malaria in terms of their T-cell response. To really understand what responses are different and which are important for protecting from clinical disease. And so to do this, we've used those whole parasite approaches. So re-stimulating peripheral blood mononuclear cells for a short time with infected red blood cells, merozoites, and cytomegalovirus peptides. And this is because cytomegalovirus is known to elicit a really strong and protective CD4 memory response that's long-lived. As I mentioned, we think opsonization is a really important immune correlate, so we've included opsonizing these two different parasites with autologous plasma. And then we're reading out T-cell effector functions as the production of pro-inflammatory cytokines, interferon gamma, TNF, IL-10, and IL-2, which is quite a Th1 biased response, but each of these cytokines has been sort of quite thoroughly studied in malaria on its own. These are the cytokines that are elicited in response to parasites. We're also looking at expression of the activation marker CD154 or CD40 ligand, and also production of upregulation of CD107A or LAMP1. And in the conference that I just got back from, there's been a lot of interest looking at these two markers, especially in some of the vaccination studies, as being possible protective correlates of effective vaccination. So we're really interested to see how these two markers play out in our population studies. And we're really focusing on polyfunctional cells, which is the ability of cells to produce multiple cytokines, as these have been shown to be really important for a protective viral immune response. So we've used this 14-colour antibody panel to address these 3 different memory cell populations. And this has really given us a lot of data to deal with. So by looking at all the different combinations of these markers, we have 17 different response variables for the different populations across the 5 different stimulation inputs. So it's a lot of variables to deal with, and we're going to be performing some multidimensional scaling analysis with the Mueller lab and bioinformatics to really kind of tease apart some of the patterns. So for the purpose of this talk today, I'm just going to show some preliminary analysis looking at the differences in responses to cytomegalovirus and malaria parasites. So when we looked at both the frequency and the proportion of individuals that responded, we saw that responses were much lower to infected red blood cells and merozoites in terms of cytokine production. When we split this into the individual cytokines, we saw less interferon gamma, markedly less TNF and IL-2, and a small bit less IL-10 in response to infected red blood cells. We were interested to see that it was infected red blood cells that upregulated the most CD107A and CD154 in response. But we only saw this for infected red blood cells and not merozoites. As I mentioned, we were interested in looking at polyfunctional responses. So when we looked here for production of interferon gamma, TNF, and IL-10, I didn't include IL-2 because, as you would have seen on the previous slide, there really were very few responses. That the response was predominantly an interferon gamma monoproduction across both the peptide and parasite inputs. But then if we focus in here on the polyfunctional responses, we can see that, again, there's less polyfunctional cells in response to malaria stimulation rather than cytomegalovirus peptide. So in summary, from our preliminary analysis, we've seen lower frequency of cytokine responses to malaria parasites than CMV, and that it's a predominantly interferon gamma monofunctional cell that responds, and that there are fewer malaria-specific polyfunctional cells. So we're really interested in looking further at these and sort of subsetting down to the level of the different memory cells that are responding to really kind of understand whether or not any of these responses are protective, and to kind of address what is the difference, and why is there a difference between the response to CMV and malaria parasites. Some other observations that we've made is that if you opsonize parasites, you have reduced IL-10 and increased CD154. And this is important because obviously in vivo, these parasites are able to be opsonized by those circulating antibodies. Obviously, there's a lot of work still to be done. We need to identify the different memory populations that are responding to each of the parasite inputs, and to really understand the pattern of responses to malaria parasites, and to try and understand if we can find any protective correlates of immunity. So just in summary, I've shown you today that opsonizing antibodies seem to be a really important mechanism of protective immunity, and that we think that understanding T-cell immunity will also be really important. It's the production of these pro-inflammatory cytokines by the T cells that can activate the phagocytes to increase phagocytosis and promote parasite killing. But it's also the uptake of these parasites and the presentation of antigens, which is really important for kickstarting this T-cell memory response. So I hope you can see that everything is quite interconnected, and I think you really need to look at both arms of the immune system to understand malaria fully. So that just leads me to thank all the people that have contributed. First of all, thanking the children, guardians and communities in Papua New Guinea. I'd like to thank my supervisors, Louis Schofield and Alan Cowman, for allowing me to work on these really fascinating cohort studies and giving me the freedom to take things where they did. Diana Hansen for a lot of support with the opsonization project and for mentoring. Ivo Mueller and his statistician Connie Lee-Wesun for statistics. Couldn't have got where we got with the analysis without them. A lot of this work was a direct collaboration with Emily Erickson, postdoc in the Schofield lab. And yeah, obviously she could equally be up here presenting a lot of the T-cell work. So big thanks to Emily. Present and past members of the Schofield lab for being a great lab to work in. The infection and immunity division as a whole. I came to WeHi 3.5 years ago without ever seeing a malaria parasite. So if it wasn't for all the mentoring and advice from many different people in the division, it wouldn't have been possible. In particular, I'd like to thank Abby Harrison for help genotyping the parasites, Chris Chu and Clara Lynn for the MSP-DVL1 antibodies, and Alex Uboli for all of his work generating the MSP3 family knockout parasites. Axel and Mark for their advice on my PhD committee. These are obviously very facts-intense projects, so the Facts Lab. I'd like to thank Doug for supporting Sophonius Tesema and I to visit PNG recently. It was really great to put our research in perspective. Obviously, the WeHi Students Association and students for just making this such a fun place to do a PhD. Danny Wilson, who was in Alan Cowman's lab, has really continued to be a great support working with these PNG isolates. These studies obviously build on a lot of work done by a lot of other people. We couldn't answer these questions about immunity if we didn't have all of the infections genotyped. And so big thanks to PNG IMR and Swiss TPH for all of their work in genotyping. I'd like to thank University of Melbourne for my scholarship and these different organisations for supporting me with various travel awards throughout my PhD. Big thanks to family and friends, and just want to mention my grandma who passed away this year. Okay.
Questions?
Great talk, Danica. I was wondering whether there are two different models of how the opsonizing antibodies are acquired. I was wondering if you could distinguish between those two by looking at the diversity of the parasites in an infected individual as they get older. You would predict that the diversity would decrease if they're accumulating those antibodies to different strains.
Yeah, that's an interesting idea. I think the issue is if you develop immunity, you still have parasites. You're just able to control the diversity. So I think if you took like a PCR-based approach to look at the diversity, I think these people might have developed immunity to those parasites, and you'd be able to detect them by PCR. So yeah, it'd be an interesting approach, but I'm not sure if it would actually read out in in the blood of these kids.
Yeah, so Danica, in your phagocytosis assay, your use of knockout parasites nicely showed that none of those single candidate antigens was responsible. But just stepping back from that a little bit, I know this is technically really difficult with patient plasma samples, but is there anything in your data or an easy way to get at whether that's driven by a few high affinity antibodies or really antigenically broad, highly polyclonal, but maybe not so high affinity kind of, can you tease anything out about the sort of broad qualities of the antibody response?
I mean, that's definitely been something we've thought about. I guess we think that these are relatively high affinity in that we literally add the antibodies, add the cells, and then they go and do their thing. So maybe some of the low affinity antibodies that might take, say, an hour at 37 to bind on a ELISA plate might not bind. It's quite tricky. When I've looked at relating the responses to opsonization to a lot of the serological work that's been done with these study samples, everything correlates. So it's, at least from some of that the specific antigen serology, there doesn't seem to be one distinct protein that might be contributing to this. I guess maybe the only thing I can think is because I do go out to 1 in 120,000, that whatever is binding might have to be pretty high affinity to make it through. But it is tricky, and I haven't definitely come up with a way to address which might be playing a role.
Any further questions?
Well, I'd just like to... Aaron.
Sorry, I'd like to follow up on that. Wondering, just with what you were saying now, how tricky it is. There's been sort of techniques using T-cell affinity that you elute the peptides off of the MHC, whether maybe some of that might be usable, not by you, but in a future approach that you actually elute the antibody off of I guess it would be opsonized, but...
Yeah, I mean, that's something I've thought about as well, sort of mixing merozoites with these antibodies and then sort of spinning out the merozoites and trying to see what they bind to. It's definitely possible. Again, it's which of those antibodies would actually result in phagocytosis is kind of tricky as well, because we do know that merozoites are really sticky. But there are lots of different ways we could try and ask questions, and I kind of think if we sort of find similar answers lots of different ways, we can maybe become a little bit more confident about that we might be finding something that's really, really relevant. But I think at least from the knockout stuff that I feel that there are probably lots and lots of different targets of this antibody response. But I guess the other question is whether or not you really need to target all of the antigens to be protected. We still don't know that.
Does anybody have a laboratory raised monoclonal that works in your assay? Have you ever, something that you could benchmark?
Yeah.
Plasma samples?
Yeah. No, that would be a great resource. Yeah. The problem being that the phagocytosis is driven by the Fc receptor interactions.
Is that cross-reactive between lots of them?
There are people that report you can do it with rabbit antibodies, but I have tried so many different ones and it does not work. So, I mean, if someone can make a humanized monoclonal, then that'd be great. But yeah, short of doing this affinity purified antibody-based thing, it's tricky. I guess most of my positive controls has just been hyperimmune adult plasma. But yeah, it's definitely one limitation of doing a functional assay, that you can't rely on things made in mice.
The movies you showed were great, very conclusive, but I was struck by the fact that you were using didium bromide to label the parasites. Has anybody come up with a method for getting fluorescence without using didium bromide? It doesn't strike me as the safest way to go forward.
I sort of played around with that at the beginning because I was like, didium bromide. There are GFP parasites but at least using the sort of episomal expression that the parasites have that have been generated in the division, you don't get enough copies of GFP. They're just not bright enough to resolve that difference between the monocytes and the merozoites. I've tried using things like CFSE and FITSE. However, because they're protein stains, they tend to sort of be quite sticky. So the background just goes crazy. So yeah, after sort of playing around with a lot of different things, I opted for ethidium bromide and just being very careful.
I'd just like to point out, I mean, I think Danica always gives a very polished presentation and we can sit there and it's a really comprehensive story, but I'd just like to stress for the audience to realise that she's developed an assay that didn't exist before, that's applicable in high throughput to current vaccine studies. She's taken it, applied it in a distinctive way to a whole bunch of parasites. I mean, endless numbers of parasites nobody ever does in complex longitudinal cohort studies. So she's essentially given you half of her PhD because the T-cell stuff is yet to come. So I think it's testament to a really extraordinary work ethic and dedication. There'll be more coming out of it. So congratulations on an excellent seminar.